Hello.
Hello. Sorry for the delay. We're here. Um, yeah, welcome to Philosophising.
My name is Ruby,
I'm Nathan.
and we're going to talk about the history of PCR.
Yeah, and I feel like we should also like talk about what PCR is. Because I think it's like oh sorry, I'm just checking this sound. Um
It's a really common tool in nearly every molecular biology science lab. I would
yeah.
say, like, this is one of the first assays you learn to do in undergrad, like, bio lab, maybe? Like, I think that's my first PCR was in undergrad biology lab class.
Yeah. I mean, I think most people also know it because it's the thing where from like 2020 to 2023 or so, like if you had to like go get your nose swabbed to check if you had COVID, and if you did the test or it was like you did the swab and then you sent it off to a lab somewhere and you got the results back, um,
Oh,
like
yeah, no, like,
a day
you
later.
got PCR tested,
That
yeah.
was yeah, I feel like that's where like most people like have actually heard the term PCR now is like in the context of PCR testing. Um,
That's fair, and antigen testing too, like, I feel
mhm, yeah,
like these, like, very I guess before COVID pandemic, was
hmm
it very less common to know as a layperson, but now every layperson knows, or at least has heard of it before.
yeah. I was I was I saw this, I think it was actually an XKCD comic where it was talking about how kind of like you can kind of infer when disasters happened by like when certain words became like public knowledge. Like when you look up like the Google spikes for like the Google searches for like European Union, you see a big spike around Brexit, or like when like that a volcano in Iceland happened, you see a whole bunch of searches for like some term about like the volcanic ash. And I feel like that era, it was just like, okay, I guess everyone has to learn about like PCR testing. Um,
Revive says glorious years when
yeah,
everyone knew the acronym PCR.
yeah, exactly. I just think it's really interesting because it went from being like this obscure lab technique to this thing that was kind of central to society. Um, and I mean, I know like when the pandemic was actually starting, like in Chicago, they were actually going around to labs with people doing um, labs with people who did molecular biology stuff, and they were basically like drafting people and being like, "Can you come work in our public health lab to help us run all these PCR tests we need to do?"
Yeah.
Um,
I feel like it's almost we take it for granted that it exists.
mhm.
Like, I just learned it. I learned how to use PCR well before I knew any of the history and, like, how the reagents came to be, like who came up with it, and I didn't realize how young it was until we were making this episode, and I was just like, "Wait, this was like cutting edge technology the year I was born? What?"
Wait, really?
Yeah,
Huh.
like the yeah, we'll go through the history and like the timeline and years throughout this episode, but like I was genuinely surprised like how young it was. And actually it makes sense. It makes sense that like this is the timeline for it. Um but like I just didn't think of it because it's just like it's like a hidden thing. It's not super hidden. It's just like you don't think about it because it's always there. It's like you reached for your mouse and you expect it to be there and it does the thing it does. And that's how common this tool is. It
Yeah. And I think like I mean I think like for most people I think like you just don't really think about it. Like it's more of a thing where okay, you go and get some sort of clinical test and like you send off a sample and it comes back.
Yeah.
And
I mean, practically in the lab, let's say, like, I have a new genotype of mice that I'm interested in. I look up, like, I literally pull up the sequence, like, design primers, and then, like, order it, and it'll show up in like a day, and it's like $5. And it took 12 seconds to put it into the computer and press enter send, and it's like $1 per nucleotide, like no, it's like cents per nucleotide, and then it shows up, and then I just mix it, and then like put it in the thermocycler, run it in a gel, and then I have my answer, and it like everything works.
And
Like
you
you
get
don't.
a bunch of mouse DNA.
Oh yeah, and obviously you start
Yeah.
off with some mouse
Hmm.
DNA, but like it's such a common thing that oh, my screen just died. Hold
Oh,
on.
um,
And we're back.
that's weird. Yeah.
Yeah, that is
That's
weird.
such a common thing in the molecular labs that we don't
Yeah.
really think about it that much. I
Yeah.
feel like I feel like one thing we should do is we should explain like, okay, what exactly is this? And so essentially what PCR is it's a way of making copies of a stretch of DNA. And so I was like, when I first started learning about genetics, I think one of the things I honestly was really confused about was that like I kind of assumed that the way that we did any sort of like genetic analysis was just you got DNA, you extracted DNA and you sequence it because like I knew like okay we had sequenced the human genome and that seemed like the kind of the logical thing that you would do. And it turns out that's usually not what you do. In part because sequencing DNA is really expensive, but also that you have basically this um, kind of bottleneck, which is that if you have a bunch of cells that have DNA in them, there's still not very much DNA in this solution. So most of the time, if you want to do anything with the DNA, you have to make a lot more copies of it before you can do anything with it. And so essentially what PCR is a way of making copies of the DNA. So you actually, and we're going to talk about how this works, but you essentially take some of the enzymes that are naturally used to copy DNA during cell division, you put them in a test tube along with your DNA and primers that essentially tell these enzymes which stretch of the DNA to copy. You put them in there, they make a bunch of copies of the DNA, and then you have a ton of DNA that's enough to analyze. And so people do a few different things with this. Actually, so one of the most common things that people will do with it is that if you want to see, like, for example, is there a specific pathogen or a specific kind of cell present in your solution, you know that thing has a certain sequence of DNA. So you make primers that will target that specific sequence. And if that sequence is present, they will bind to it and they will make a bunch of copies of it. And if it's not present, they won't do anything. And the interesting thing is that you can do this and you can you can essentially use this to detect whether that thing is present. So like when we're testing for COVID or other kinds of infectious disease tests, we're targeting a part of the genome and we just make primers that make a bunch of copies of it. If you make enough copies of it, your solution actually gets so full of DNA that like its optical properties change and it gets like gooier, and then you can detect it. But if that sequence is there, never there to copy in the first place, nothing happens. And so that's called quantitative PCR. And the idea is that you can use that to say was this present in the solution in the first place. And you can use it even when you have maybe just like one virus, like something that would just have way too little concentration to be detectable without doing this kind of amplification step.
Yeah. So
Um,
you can kind of know by how many rounds. So like every round you have one like annealing step, like one like it's like one replication round, and so you can guess, you can like back calculate how many numbers of particles of that DNA sequence existed at the beginning. So you can kind of be like, hey, like, do you have a lot of this particular gene in this sample, or do you have a very small amount? And that's the beauty of the quantitative. So like they basically replicate it one round, and then they like image it real quick to see how the optical properties, and then they replicate it again, and then you measure it again, and you can basically create a graph of how many like how much optical diffraction happens, and or they used a dye to see how much of it existed, and then you can calculate how much you started off with, and it was like, hey, like the sample had this much of that DNA in it to begin with. um
right. Because
Another.
you can kind of measure how long it takes to get to a detectable amount of it.
Yeah, another really useful use for PCR is if you know there's like a mutation, for example, and this mutation, caused a frame shift or like a loss of a couple of a couple of base pairs. You can basically put the forward and reverse primers and then like cut these sections, and then if there's a mutation, it'll be either longer or shorter than the not
Mhm.
the healthy version of the gene. And that's actually how they found diseases with lots of replicating repeats, which I talked about previously in this on this stream.
Because it
Um,
only works like so it only works if it's like a big mutation, right? Like so if it's just like a single base pair change, I guess you can't detect it with
you
that
actually a
technique.
single base pair might be really rough, but you can do three. And so the way to do that is like for once you make this massive amount of replicating like of one amount of segment of DNA, what you do is usually run it through a gel. And so DNA has like a positive and a negative side. And so if you use electrolysis, you can like pull it through like a matrix, and then like the small ones will travel further than the big ones will. And then you just run it really long time, and you'll see which one is smaller. And you
But like so it doesn't work like if you have a mutation where it's like there used to be a T and now it's a like C or
uh
something, right?
yeah, you would design a different experiment for that. So if you want to figure out, so you can like do really clever things with primers where you chop it up in like different sections, and then you would look for the length of the sections and where they like
Hmm.
all the different so you can do like restriction fragments where you like restriction enzyme a whole bunch of shapes, and then you can find out where it didn't cut the exact same way, and you're like, oh, it must be between a C and a T or something, and then you go to all those sections and you do it again, and you go before it and after it, and you kind of narrow it down that way. And it's like process of elimination. Yeah, but anyway, that's a cheaper way to do. Besides, I mean, the old school sequencing was also that way, but like this is how a quick way to find where something changed. And you always have to compare it with a control, right?
Hmm.
We're
Yeah.
getting ahead of ourselves. Um,
Yeah.
the idea is that we're just trying to replicate. PCR allows you to replicate a chunk of DNA pretty reliably and make a lot of it, so that we can actually work with it. And so, I guess in the layperson's world, like CSI world, let's say there's like DNA left over from a crime scene, like you have to amplify that little amount of DNA before you can like identify who done it. Does that?
Right. Like I
That's
guess like
probably like
one yeah.
a
And
more
like I think like
layperson.
it's like I guess the way I think about it is like so one thing you can use it you can use it to kind of like see something about the sequence of the DNA or kind of see like what patterns are present in the DNA by doing this kind of selective amplification of it. The other thing that you can do with it, like the other reason people amplify DNA is to actually do genetic engineering. So, for example, like if you find say a particular kind of cancer cell that seems to have some odd mutation, maybe you want to put that gene into some different cells so that you can check whether that mutation is actually what's making your cells weird. And like again, you need a lot of DNA to be able to do that. So what you'll do is you'll design the primers to replicate the area around the gene that you think is interesting, and you'll just use that to make a ton of copies of that DNA. Sometimes you'll then insert those into some other DNA that helps them get integrated into the genome. But in some cases, especially with bacteria, you can just kind of take the DNA, replicate it, and then you just have your bacteria kind of like swimming in this like soup of amplified DNA, and then you just like poke holes in them, and then enough of the DNA gets into the bacteria that it starts getting transcribed. And that's how you can basically genetically alter these bacteria.
Yep. Genetic
So yeah, so
engineering.
this ability to like make copies of the DNA, it turns out is basically like fundamental to all modern biotechnology. Both if
Yeah,
we want
almost
to
like
see like what
modern
is something,
biology.
what is this
Yeah.
DNA, we want to edit the genes and do something else with them.
Yeah. Um, so historically, like the idea that you could do this, do PCR at all. I mean, first we needed to know like the structure of DNA. So like Watson, Crick, Franklin era. Then we needed to figure out like polymerase, which is the enzyme that allows basically to take to find the correct like to like oh man, I do not have my words today. It's Friday afternoon. Polymerase basically like helps the correct amino acids like bind to the parts that are open. So and then you needed a way to like cause the DNA to denature. I mean, sorry, not denature, to like unzip and then rezip correctly. So you need to like unzip it and then like get things to bind onto it. You run the polymerase across so that it like makes the thing, and then it has to like reanneal back to stick out together. And so we had figured out like temperature could cause the like the unzipping and then the rezipping. Oh my gosh, my words. Annealing, that's the word.
But I think rezipping is good because it's basically like when the DNA gets up, so the DNA is a double helix, right?
Yeah.
And when it gets above a certain temperature, the two halves of the helix actually just separate from each other. And that's important because you need to do that in order to make a copy of one of those DNA strands. So there
Yeah.
you're going to make a copy that binds to half, like you have two single helices and you're going to use them as templates to build your new DNA strand. So you unzip them from each other, then you can build the copy.
Yeah. The next thing had to happen was that you needed to know that you need to have DNA templates. So you need to know what DNA you're gonna amplify. So you need primers that would like
Mhm.
grab the front end and the back end, like a forward and a reverse, to figure out like where like between the section between the forward and reverse primers or where you're going to amplify like with the part of DNA. So we need to figure all those parts out. And so all of these things were kind of being worked out by molecular biologists and chemists and in like the 50s, 1950s, late 50s. Um, and it was very slow. Like for example, like heating like the DNA to get it to like unzip would take like 2 minutes, like full 2 minutes just to like heat them and get it to do the thing. And then you had to like chill it back and then like add the DNT. Oh, sorry. The components of a PCR reaction are the DNA template, the primers that go both directions, the polymerase, which takes like allows the DNA to like attach the correct amino acids to the unzipped DNA, the DNTPs, which are literally the amino acids like hanging out in it, buffers and dyes, so you can like see what you're doing once it's amplified, and then you also need a little bit of magnesium because it helps with the binding, and you need like a water bath and like cooler or thermocycler to like basically change the temperature that this little amount of solution is going into. So those are the components, like the bare minimum components you need to like run a PCR reaction. And even getting all the parts in the right place, like it took us until like the 70s. Like the
Wow. Yeah.
1970s, like they were doing this really slowly. It would take a like a postdoc 2 years. I wrote this down because I was like, it took them 2 years to synthesize just the primers, just the forward and reverse primers that are like the correct number of like they're targeting a specific gene.
So
And
it was like the generating, like just making these DNA primers that was the limiting step.
yeah, and it would, and they were organic chemists, so they would have to
Mhm.
do like these, bonding things, and then like extract, and then bond, and then make sure actually the correct thing got extracted, and then like put a lot of what they extracted back into a different reaction, and then try to like bond the next nucleotide onto it. Like they were doing it like one nucleotide at a time, like one chemical link at a time, and do all kinds of like crazy like chemical maneuvering just to like make these primers. And it was like so valuable, like the labs that worked on this would keep their like one or two samples of it like under lock and key, and two people needed a key to open it. Like I was just like reading
Wow.
these accounts of like this is like, oh, did I write the name of the labs that were doing this down? I should have. um yeah, but like that's how valuable it was. And like like the postdocs that wanted to work on it would be like, please, we really need like this primer. You would have to like petition to use it. So anyway, large scale amplification of DNA was just like, it was a pipe dream, like in the 60s. That's not long ago. Like our parents were alive,
Mhm.
but like it's not that long ago when it was like completely a pipe dream to do large scale amounts of DNA replication. um And then let's see. Oh, and so they have, like, during this time they were describing possible ways to do it. And so the Klepi lab, Klepi and Corona lab, who in 1968 did describe the first version of, like, if they could do it, this would be perfect. Because then they did do really slow versions of this in their lab, like 1 or 2 reactions. Imagine like replicating it twice and being like, we did it. That took us 2.5 years.
Like,
We
okay,
replicated
done.
GNA
Yeah,
twice.
we did the
Two
did the
cycles. And so that
yeah.
was the first published like description of the technique. And I'm going to read exactly what they called it. So this was published in 1971 by Klepi et al. The principles for extensive synthesis of duplexed tRNA genes which emerge from their present work are the following. The DNA duplex would be denatured to form single strands. This denaturation step would be carried out in the presence of sufficiently large excess of the two appropriate primers. Upon cooling, one would hope to obtain two structures, each containing the full length of the template strand, appropriately complexed with the primer. The DNA polymerase will be added to complete the process of pair replication. Two molecules of the original duplex should result. And the whole cycle would be repeated, and there will be added every time a fresh dose of all of the enzymes. So,
Mhm.
uh, it was
So
described
basically, so
first.
you heat this thing to unzip the DNA, then you let it cool down so it can start making the new copy, and then you unzip the new copy from it, and then you put in more enzymes
Yeah.
so that you can then make copies from those.
Yeah. And so at this time, they were like, this is technically really difficult to do because you got to do all these like water bath situations. Then you need to re-add all the enzymes, the polymerase enzyme, and then you also had to synthesize all the primers, the oligonucleotide primers, which took 2 years, by for example, to make a 10 length 10 oligonucleotide length primer. And it just took a really long time, and you can just replicate it like twice, and they're like, theoretically could do it. This is a description of how to do it if you were to do it. Meanwhile, so in the 1960s there was I think he's a microbiologist in Indiana University, Thomas Brock, who was interested in studying microbial ecology, and he started studying the microbes in tidal pools, freshwater lakes, spring lakes, and geysers and hot springs. And so he was on vacation, I think, and he had to travel, and he traveled to Yellowstone National Park, and he like grabbed some of the samples there, and specifically at this hot spring called Octopus Hot Spring, and the temperatures of Octopus Hot Springs was 80 degrees, and it fluctuates. And that's actually where we get the first example of Thermus aquaticus, which we will call TAC, because the T
Hmm.
for
Mhm.
thermus, and aquaticus AQ is the TAC. And so that's where TAC comes from. So TAC is a polymerase that actually is stable under fluctuating temperatures. So instead of having to add the enzyme in every time, you could just put in some TAC, and TAC would survive. So you could just change the
So you
temperature
can
again.
heat and cool it multiple times without having to put in more.
Yeah, and actually, I ended up going through a rabbit hole of what the Octopus Spring came up. There's actually like several hot springs in Yellowstone National Park that are like some of the most heavily studied springs in the world, and like it has really cool things. But let me find the description of of TAC when they first um extracted it. It was like a pink filament. Oh yeah, so a pink bacterial filament living in the hot spring. So it was like he grabbed some of it, but actually it turns out TAC actually exists in a lot of different spaces where the temperature fluctuates. So for example, you can find it in hot springs in around the world, in Japan, New Zealand, Iceland. It's also found in like soils in tropical areas where the temperature fluctuates really like to really hot temperatures and then comes back down. And it was even found in the hot water supply in Indiana University. So it's actually
Oh
like
wow.
not a
Uh-huh.
bacteria that only exists in the hot springs of
Yeah, it's not a
Yellowstone National
rare
Park.
bacterium.
Yeah, it
Yeah.
actually exists in other places, but like that first TAC was, identified in this geyser. Anyway, um,
And so who is the person who found the tech?
this guy named Thomas Brock.
Thomas Brock. Yeah.
Yeah, and he
Mhm.
published it, and he talked about it a bunch at conferences and whatever, and it was kind of not really, it wasn't a big deal until people were
Hmm.
trying to do PCR. And then they're
Hmm.
like, hey, like let's combo this. And so that actually brings us to, I guess, the most famous person that people talk about when they're like, oh, the PCR guy. That's
Mhm.
Mullins.
Mhm.
So I think he was working at a he was his PhD is from Berkeley, I think, and he was working in industry in Emeryville in this place called, oh, what is this company called? It starts with a C. I wrote it down. It's called
Beaches.
yeah, Cetus. Is that right?
Mhm.
Yeah,
CETUS. Yeah.
Cetus. Yeah, so
Uh-huh.
he was like a staff scientist there. Kind of wild story. But he, at the time, he was working on amplifying DNA, and he put together this method where you can combine the temperature, the TAC polymerase, which is not which can be stable under what range of temperature, and then he basically put all the components together and like showed that it worked. Which is like really exciting because like it exists, and actually for a while people didn't believe it worked because he showed it to people in the building or in at work, and people were like, wait, what? But once it was finally like proven to work well, they awarded him like $100,000, and I was like, congratulations, you did your job. And then they like sold the patent to it for like $300 million. And I
I was
think that was like where he like we broke him. I
That was
think.
where he lost it.
Yeah.
Um, I don't know.
And
I mean, okay, I'm reading this Wikipedia article on him, and it does say he acquired a reputation for erratic behavior at CETUS, once threatening to bring a gun to work, and then also engaged in public lovers' quarrels with his then girlfriend, and nearly came to blows with another scientist at a staff party. Um,
yeah, he this man was a wild child. He
like I
had
feel like he had problems before he discovered the PCR. Is what I'm thinking.
um yeah, they did award him
I
the
was
Nobel
curious though,
Prize
like,
eventually.
yeah. Oh, he did get a Nobel?
He did. The Nobel Prize, I forgot which year, prize which year. Um PCR. He got it in, it's recent too. It's Carrie Mullins in 1993.
Hmm.
Yeah, like I was a year old when this
Wow.
happened. I
Yeah,
guess
yeah.
in my mind it's like, oh, it's not that far ago because I was alive. Um, um, I mean, this man is very problematic in many ways. And
Yeah.
also, I mean, he wasn't the first person to describe PCR. Like it was already described before. He was able to implement it in large scale. I think
So
that's
he
the
was so he was able to get it to work more efficiently than the previous people?
yeah,
Is it
like
like
yeah, because like I mean,
Mhm.
before this, like we didn't have all the components to do
Mhm.
this quickly. Like it took ages
Mhm.
to make oligonucleotides. I think now it takes like a machine to do it really quick. Um, and he correctly found a polymerase that worked in extreme temperatures, and they were working on a project which like needed like amplified amounts of DNA, and so he like reliably did it a lot in
Mhm.
industry.
That makes sense. Yeah. So he figured out like so it wasn't just the temperature thing, but he figured out some of the other technical challenges in this.
Yeah.
Yeah.
Um yeah, and he like he did, I think, oh yeah, and actually okay, to be fair, he also was unaware of the descriptions that Klepi and Corona had made. Like so he
Mhm.
was not he didn't read their papers
Their
before
paper.
he put it together. So it was like convergent thought. It's kind of like how the
Right.
radio was
Yeah.
invented
Yeah.
around the same time by like multiple people that weren't aware they were also working on the radio. It was just like
Hmm.
around that time, like there was just enough tools, and then like people wanted to look for it. Like it was,
Yeah. Like a lot of people are just thinking like, okay, like why not use these DNA replication mechanisms to um
yeah.
amplify the DNA?
Yeah, I actually find the story about how TAC was found really cool because,
Uh-huh.
like, yeah. So at the time, Brock, who was the biologist who from Indiana University, that was like, I'm gonna go check Yellowstone, he actually didn't really want to go Yellowstone because he thought it was too filled with tourists. Um and luckily, because it was a national park, it was actually not developed into, it was a thermal pool that wasn't developed into like a hot spring for vacationers. Like no humans were disturbing it,
Mhm.
and it actually really preserved the microflora in that
Oh,
spring.
interesting. That makes sense. Yeah.
Yeah, and it was like a whole thing because like it's like, I mean, these pools are studied because they have interesting bacteria and they're super useful to humanity. And like if we had made them into, like a vacation pool where people would just splash in them, like we could have disturbed the microbiota and not have tac. Like imagine if we didn't have PCR because we wanted another sauna, outdoor sauna
Yeah.
vacation. Do you know what I mean? Like it could have ended up being
Like
a
it could have ended
tourism.
up just being not a thing. Yeah.
Yeah,
This
if we had
actually
not saved the land because it was a
this,
national park. Yeah.
so this is an interesting thing that I ended up reading about. So I was reading some of the things about the history of this, and I stumbled onto this one part that made an interesting point where it said after so it's talking about the patent. So it said so he got 10,000 dollars. Roach patented immensely from it with annual PCR sales reaching 5.4 billion in 2022. And then it says, despite these profits, neither the National Park Service, Yellowstone National Park, or the state of Wyoming have received any share of those revenues. And like that actually made me really curious because I was like, wait, like is that a thing that typically happens? That like if you find something in a national park that like the national park gets a royalty for it? And it turns out it did not then. So then at that time there was no like revenue sharing thing, but actually now like if you kind of have a permit to do research in a national park, they actually do have this revenue sharing program that where the National Park Service gets a cut of it. Um,
Yeah. It's weird
which I think
because
makes sense. I mean, like when you think about it, it's like, okay, the National Park Service is doing a lot of, I mean, they are providing the kind of security and safety and access to these pools that let people do that kind of research.
yeah, I was gonna say that like while tac, like the bacterium,
Mhm.
like exists in thermal pools around the world, like it wouldn't have been discovered had it not been in this space, right? Like you could be swimming in tac in a hot pool in Japan, right? Like you could
Right.
be doing
Uh-huh.
that, but a scientist wouldn't have gone and collected it from the hot
Yeah,
pool where some dude is feet is also in. like it's
exactly. Yeah. And I mean, I think in general, like, I mean, like the kind of the role of the Park Service is like, okay, you have this exotic like ecosystem where you have all these bacteria living in the water because the water gets extremely hot. So anything that can survive in there needs to have like a very distinct biology. It's like I think it's interesting. Like, I mean, the role of the park system is like, okay, they built a road so that people can go and like get these bacteria and also keep it like isolated and like as undisturbed as possible so that like those bacteria continue surviving and they provide like the safety services so that like you can if you can go in and get back the bacteria and not just like die from inhaling toxic gas, that sort of thing.
yeah.
um,
No, I was gonna say the springs are currently still under investigation. There's like
mhm,
several springs that are like in that near vicinity in the lower basin part of Yellowstone National Park, and they have like different amounts of oxygenation level. And so like the Octopus
mhm,
Pond has like a low oxygenation level, and there's like two ponds near ponds. That's not the right word. Geysers, springs, hot springs that are near it that have different levels of oxygen and like different amounts of fluctuation of temperatures, and like each one of those are completely different ecosystems. So it's not like we can't just save like one pool. Like we gotta save all of them. Like this is part of like this is an example, like a clear example why like we need to conserve like
yeah,
nature.
although it's like slightly undermined by the fact that the like the tack was also just like in the tap water when you looked at it, but Um,
but in such small quantities that like
mhm.
you wouldn't have isolated it, right? Like this was
I
like the
see. Yeah.
overwhelming majority like of this pink filamentous thing they grabbed was like this
Mhm.
thing, right? Like
Right,
you
where
could
it's like
probably
there's actually like tentacles of it growing where
yeah.
you can be like, what's this?
Yeah,
Um,
whereas in like nature and in the soil somewhere, there's like much less of it. I mean, in general, like nature will do its thing, so it'll survive. Even
mhm.
I mean, there's bacterias that can eat like styrofoam now, so like whatever environment we give it, it'll survive. But here is like a clear example where something is really important to humanity and like science, and it would never have been discovered had we had fully overdeveloped. And we don't know what the next discovery is going to be out
Oh
of
yeah.
those pools or any part of nature that we adulterate in the future. So
Ribeye B says, gotta check in my water boiler, and I'm assuming to like look for weird bacteria in there.
so I
Uh,
actually learned about this when I was one of the first labs I ever worked in when I was in California in high school, and I was learning like just doing genotyping for someone in school, and she one of the postdocs that like was training me said, oh yeah, we ran out. I was like, we ran out of tack, and she was just like, oh, don't worry, I made some homemade tack. And I was like, what? She was just like, yeah, there's some homemade tack in the freezer in like a 15 mil tube, like just go get it.
next
And
to the sandwiches,
yeah,
like
I
yeah.
was like, homemade tack. And I was just like, what do you mean homemade tack? She was like, oh yeah, I isolated it myself. And I was just like, where did you isolate it from? And now I realize like she had probably isolated it from like the water bath or something in the lab. And she was just like, yeah, it was like a side quest at some point. And she was like, yeah, it works just as good as the commercial tack and it's free and I have so much of it. And I was like, okay. But it literally said like homemade tack on it. And I was like, oh, okay.
You're just like, huh, and it worked. Yeah,
Yeah, yeah, it totally worked. Like
yeah,
there was no problem with it. But like I didn't realize you could just make tack. And she was like, yeah,
yeah.
once you know what it is, you can isolate it. Like I was just like, oh, that's cool. That makes sense. Um,
Interestingly, this was also when I worked in a lab in high school. We also had a similar discussion where the people were like, yeah, we can buy this tack because it's really expensive, or we can just like grow a whole ton of it and freeze it.
yeah.
Um, so I guess, and so apparently, like, I mean, so tech is still like kind of expensive. Its price has come down a lot, especially for like specialized varieties of it that are under patent. Like there's varieties that are like especially accurate at copying the DNA and stuff. And it can actually be quite expensive. Like it can cost like $1,000 for a little vial of it. And back then it was even more expensive. So people were just like, why not make it
It
ourselves?
was worth it. I feel like in 2000, I guess 2010 era, that was when my I was being trained how to do this and they were
Hmm.
making homemade tech. It was worth
Yep.
it to just make it themselves. I was gonna say like now they're selling kits. Like when I was learning how to do PCR reactions, we would like measure out every component. Like we measure out how much DNTPs, how much magnesium, and like we would do it separately depending on like the length of DNA that we're replicating and like how long the primers were, like all the different variables. You would just have a notebook where you could like get like the best reactions and like, okay, this one was really good, you didn't get a lot of like side random bands, and like you would just
Hmm.
like twiddle it and do a bunch of stuff. Nowadays I feel like you can just buy like a tube and it's like this will work on 80% of most things and you
Hmm.
can just pop in your DNA template and then like it just goes. And I was like, "Oh, okay." It's
Yeah, you're
it's
like, okay,
moved.
it's
Yeah,
like a more straightforward
yeah.
procedure now.
It's
Yeah.
really like I feel like the technology has moved so quickly in the time
Hmm.
that I like even when we began training on how to do it and how it's done now. I mean, it's just like you don't even need to think that hard anymore.
Yeah.
Like you don't need to be like, oh yeah, maybe a little. We can like bump the titration of this a little bit more, or dump bump a little bit of more magnesium, or like let's anneal a little bit longer, or like let's like take the temperature of the thermocycler, or like let's do like two more cycles, but like a little hotter at like in the like the fifth and sixth cycle. Like
And
nowadays,
is
the
that
tack is
because
so good.
we have enzymes that work better now? Is that the main reason? Or
Well, I think it's just been so dialed in and it's so useful for so many people, and they've tried to use it. They try to make it really machine friendly. Like
Hmm.
there's the artisanal parts of it have been kind of
Hmm.
like they have figured out the recipes, like really
Yeah.
robust recipes where it works in like many contexts, and they just are commercial. You can just buy it, and like you can buy it, and like it'll even have loading gel coloring it in it. Like
Hmm.
it's like one tube. You just drop your DNA in, put in the thermocycler, run it, and then like you'll magically get not magically get you'll get exactly what you asked for. Does
Uh-huh.
anyone know a better and bigger target inhibitor basis from Ki than these binding? Uh, okay, that's a very
That's a
specific
very
question.
specific question, unfortunately.
Yeah,
Um,
off the
better
top of my
target
head,
inhibitor, so it's better at inhibiting mTOR. Oh, I think there's supposed to be. Oh, from a. I don't know that, unfortunately, is
bases.
the short answer.
Yeah,
But
sorry, bases
uh,
from Ki than those binding DB underscore AI. I actually don't know what that means. Can you elaborate on your question? But anyway, we
I
can
was
keep
also gonna
powdering on.
say, I think, yeah. But anyway, we've kind of really dialed in these techniques, is what you were saying.
Yeah, like we're so dialed in, like or like companies, industries have dialed it in at like this point. It's like there you don't really have to futz with it as much anymore, which is kind of nice.
I also think like at the start when I was talking about how like okay, I assumed that like when you wanted to do stuff with DNA, you just sequence the DNA. Like we don't that's totally not really like completely a thing, but it's become increasingly a thing. So like we were talking about at the start, we were talking about at the start about like okay, you can use PCR to like look for these different mutations, and you make basically like primers that will bind to a specific sequence, or you make primers that bind to two different things and you measure how big the thing in between the primers is.
Yeah,
And people still do that, but increasingly people will also do this thing where it's just you kind of you amplify your whole area of DNA where you think mutations could be, and then you sequence them. Or you will do what's called something like RNA-seq, which is actually you just take all the RNAs of genes that are being actively turned into proteins, and you basically sequence them again using a similar technique first to like amplify them and convert them into DNA. So it still uses PCR, but instead of trying to do these like one-off assays where we're asking like, does it have this in it? Does it have this in it? Like sequencing now has gotten like good enough that oftentimes we'll just kind of be like, let's make copies of everything and then read it and then see what's in here.
yeah. I was gonna say I was kind of, you know, I didn't even think about the patents that
Hmm.
that it had because like the patents didn't expire until like 2010. Like it makes like now I understand why because like before 2010 to
Everyone is
purchase.
making their own enzymes.
Yeah, but I think now that the patent has expired, it is now truly free for all, I guess. Now we can like any company can sell tack. Like they can
Hmm.
make it and sell tack. They can make and sell, PCR, kits. Um
Yeah.
and it's such a common assay like I think high schoolers, middle schoolers will like extract DNA from strawberries and run. Do you know what I mean?
Mummies.
They'll run
Yeah.
PCRs
Hmm.
on on Amplify and whatever. Like it's so common now. I didn't even think about the limitations of patents at the time.
Yeah. And it's kind of interesting, like because this is a biological enzyme, like once you've discovered it, you can kind of just put the DNA that makes the TAC enzyme into whatever, like some bacteria, and basically you just grow the bacteria and it will make a whole bunch of this enzyme and then you can just harvest it. So it's like I think that's the really interesting thing about these molecular biology tools in general. It's like if they're enzymes or something, you it's pretty easy to just copy them even if they are under patent because like the bacteria want to replicate themselves.
Yeah. And it's kind of like I feel like this is kind of where CRISPR is heading. Like CRISPR had like a whole like patent thing and it's like a huge argument. But I think, in 10 years or whatever, it's already being used in the style of PCR, like where
Mhm.
there is no licensing. It is kind of open source amongst scientists that are like, this is a tool. Like you're gonna patent. Sorry, you're gonna
Yep.
like charge me every time I use a hammer. Like,
Is CRISPR like expensive to run? Like
I think it's expensive to make animals because to do CRISPR editing, it's mostly in cell culture and in
Mhm,
yeast and stuff. It's pretty cheap. Like,
mhm,
I mean, minus the molecular tools like dishes and media
right.
and stuff. So it's not that expensive to like incubate a bunch of cells and like do kinds of a bunch of editing in yeast and in E. coli. I think the expensive part actually comes in when you need to like shove things into animals.
Right. So like the cost is just the cost is mostly just like the base cost of the rat or
Yeah, yeah, or
yeah.
like doing in vitro fertilization or doing like
Hmm.
things like related to
Hmm.
like animal husbandry, and that's
Yep.
just expensive. Like a cage is $2 a day, right? Or more.
Yep.
So like that part makes it expensive. But like the actual CRISPR, the actually doing the maybe we should do a CRISPR version of the stream.
Oh, we can. Yeah.
Yeah.
Oh,
Um
also, Read B says if you use G0, no. I guess that's a form of CRISPR.
I don't know what G0 is. What is G0? Do you think that's an answer to the mTOR thing?
Oh, maybe. Oh no, it refers to the CRISPR gene editing technique in the first generation of an organism to
Oh.
allow the
Oh, okay,
researchers
got
to
it.
assess gene function without needing to wait for multiple generations to establish stable nucleotides.
Yeah,
That makes
that
sense.
makes sense.
I
That
feel
makes
like
sense.
people really like, I feel like the cost of lab animals is much higher than people think. Like we are in our lab are currently trying to like get rid of wild mice that keep showing up here. If you want to buy a mouse, it's like what, like $50 a mouse? Like
Sometimes it's more expensive, like more like 200 a mouse.
200 a mouse?
Yeah, it's about
Wow.
200 mouse. So like it comes out almost even if you only know exactly which mouse you're going to need, like breeding
Hmm.
versus buying. But if you don't know exactly what mouse you need, and sometimes you need the wild type controls, it's sometimes cheaper to breed.
Hmm.
Like currently, actually in my facility, I did the maths one time because they're like, "Why don't you just
Hmm.
order the mice you need?" And I was just like, "You want me to order all the mice I'm using for this experiment? It's $200 a pop." And then I was just like, "Wait, I can just breed them for free." And I was just like, "No, actually, breeding is not free. Breeding is cage space, and then if you count like 250 per cage per day, it actually
Like
comes
then
out."
it's just worth buying the mice.
Yeah, so it comes out. I think they did that calculation. Like I didn't do this calculation. I think the people that
Mm.
sell the price of the mice did this calculation and was like, "Yep, you can afford this."
And that's how they price their mice. Yeah.
Yeah,
I
because
think it's like really, I just think it's really amusing. Or I'll just be like, yep, today I spent $800 on rats. So something that most people try to get rid of.
yeah, I spent.
Yeah.
I think currently, I hope I don't get fired for this, but I spent like $30,000 on mice a year.
Wow. Yep.
Like it's like half. It's my half my salary.
Hmm.
Like I basically I'm like 150% of the cost that like when people are like, "Oh, this postdoc costs this much money." They're like 60 grand. Actually they cost 60 grand plus the materials they use, which include 30,000 for mice.
Like, how many mice would you estimate this is?
30 cages.
30. Okay.
Yeah.
So like $1,000 per mouse.
Yeah.
Or no, because there's
Per,
multiple in a cage.
per cage.
Yeah.
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 It comes
Okay,
out.
so the mice are still being paid a substantially lower salary than you are.
They are. The mice are getting paid less than I am. Ad lib food and really good healthcare.
Yeah, yeah, yeah.
For humans to go
Wait, are
and
these mice making more or less than minimum wage? I wonder. Uh,
they are sharing cages. If they were single
oh,
housed,
that's
they would
true.
be
Yeah,
making minimum wage actually.
they'd be making minimum wage. Yeah.
Yeah.
Okay. Yeah,
But we don't single house them because they
yeah.
like to be
Because they like to be
they like
with their friends.
friends. Yeah. So we
Yeah.
try to group house them so they have company.
Hmm.
Um anyway, yeah, hope I don't get in trouble for telling you all that. But yeah, animal research is really expensive and really important because we can't do many of the kinds of neuroscience experiments without them.
I like how Rabbi B is talking about the crispy injected animal.
That is
Like,
a
oh yeah,
great
that's
way.
an animal that's been CRISPR. It is a crispy animal.
I mean, CRISPR is really exciting. It's a really exciting technology because you can edit germline cells. You can edit cells that, are the progenitors of all future parts of the being. And so you can also like curtail a disease
Hmm.
that like constantly makes a protein that's bad, right? Like you can imagine if this is a disease caused by accidentally making blank. You can literally just CRISPR that part out and then they'll just stop making that protein like for the rest of their lives. That's very exciting. And there's like cool ways to like get it into the system now, like there are less painful like gene editing. um it's going to be interesting to watch how the it shakes out when it comes to like ethics and like
Hmm.
using it on humans. And I think this we're in that era now. Like that's so fast. We're
Yeah,
in like science fiction era where we're like debating like is it, okay, is this ethical to do to a child without their permission as
right.
or
And
in
people have
utero.
actually done it.
Yeah.
So there were two twins who were CRISPR edited to try to make them resistant to HIV. And it didn't really work, and it was widely condemned as unethical, but people have done it.
No.
Um,
And like this is a whole ethical thing that we need to teach people now. Like like it's a full new philosophical like conundrum
mhm.
because like the intentions are good and like I was just like this is, the beginning of a bunch of sci-fi stories, right? Where you have the best of intentions and then there was no ethics board and they didn't think it through and then they made a thing or they made a being and then we have to deal with the consequences of that, which is I think a great
I do
topic
think it's
for
like,
future
yeah,
philosophizing.
I do think it's interesting that the CRISPR, like the CRISPR or germline gene editing, like it seems like there is this very unanimous consensus that it's like, okay, this is ethically problematic because it's like you're making this person um, with this modification, and like there's all these ethical questions. I do think it's weird that we talk about that so much in the context of gene editing and not in the context of other things. Because like there's a ton of things that you can do to small children that have long-term effects on them. And it seems like very little of them are talked about in the same way.
You mean like environmental things that we could do to children that are like clearly not
Or
helpful?
yeah, I mean helpful or not helpful or like both. I think, I mean, like, there's beneficial ones, um, like folic acid during pregnancy, for example. And there's ones that are like we actually know are really harmful, like social isolation, and there's even biomedical things um, like developmental exposure to different medicines and stuff. Like, I think it's interesting, for example, like we don't think about like developmental exposure to say like statin drugs in the same ethical framework as we do like this sort of gene editing. And except when it comes to trans people where like they then everyone freaks out about it for some reason. But we don't think about like the things that like acetaminophen or statins or like it seems like there's this small handful of people like issues where people are like this is an ethical dilemma.
That's true. We do pick and choose our examples because they're easy to explain. But yeah,
Yeah.
a lot of
And I think because they feel
over the
dramatic.
counter. Yeah, and I was gonna
Yeah.
say a lot of over the counter grass, past supplements have
Yeah.
really not been tested for what they can do to anyone.
Hmm.
Okay, so Guy Noxon has come back with a more in-depth description of his question, so I'm going to read it. I am concentrating on finding the most potent mTOR protein blockers tested in vitro and in vivo written chemically in the smiles with KI. Ki, in short, is the inhibition constant that determines the strength with which a molecule blocks, inhibits the activity of a particular protein. Some databases have them, but they are terribly rare and need to be purified. I found and purified around 340
I think
XEs.
maybe KD, or I
Yeah.
don't know what the unit is.
And I
But
need a lot of them from my AI network. Interesting. Is
unfortunately, I don't work on that protein, so I
Yeah.
wouldn't.
I don't work on that protein. Uh Also like AI network. I'm with Revive on there, my dude. That is a little bit off topic. Um
If you've ever been on the Lab Rats subreddit, I feel like someone on there might know how to answer that question.
mTOR blockers.
Mhm.
I would check Tokris. I would go through the catalog of Tokris and filter. That's probably where I would start off looking for um like antagonists and agonists of stuff. Hopefully that's helpful. Sorry, we don't work with them, Tor.
Yeah, no worries. Do we
Uh,
have a um pleasant ending statement?
I have a national park story that's kind of cool. I
Ooh,
think Georgia's
uh-huh.
gonna get their first national park. I think it's
Oh
on
wow,
the
they
books.
didn't have one before.
Yeah, they didn't have one before, and they might get one. It's gonna be in Macon, Georgia, and it's a burial mound, which is kind of cool.
Hmm.
But yeah, important because more preserved land means more biodiversity,
More
and
interesting things to find. Yeah.
yeah, and actually now I wonder if I ever visited Yellowstone Park again as an adult, I should go and pay homage to these pools for offering.
I wonder if there is like anything there about PCR.
Oh, like a plaque or something.
Yeah, just like signs explaining like, oh, do you see these weird pink tentacles? They actually contain tack. Like,
Yeah, from this pool,
yeah.
uh, we have one of the greatest molecular tools in humanity. Came this polymerase that can survive
Yeah, I should
temperatures.
look this up more, but I'm curious, did he go to Yellowstone specifically to get weird bacteria, or was he just there and was he
He
just like, what is this slime?
No, he started a research station there, and he was collecting
I
from
see.
all kinds of water sources, including the
Mhm.
hot springs. So he was doing like all kinds of water, not even the springs. And he was collecting like in different places too, like not just in Yellowstone, like everywhere. He was
I
like a
have the
water
Yellowstone is where
ecologist.
he found the tack bacteria.
Yeah,
Yep. But yeah, national parks are cool. Yeah, there's
visit
lots of
them.
weird bacteria out there.
Yeah, and this is like a homegrown American American bacteria.
Tack made in the USA.
Anyway, I feel like that's a great way to end.
That's a great way. Yeah, buy American tack.
Or
Anyway,
grow your own.
yep.
Make your own homemade tack.
Yeah. Anyway, yeah, we will see